real pleasure to introduce Tony Hunter. I'm sure very little introduction is needed. Tony has been very supportive of HKUST. He is the IAS visiting senior fellow. He is currently the Dulbecco chair professor at the Salk Institute. So actually, amazingly, in one phrase, we mentioned three very important biologists. Jonas Salk discovered make polio vaccine available. Dulbecco really make tissue culture available for everybody. And Tony, of course, was a discoverer of the tyrosine kinases. And also, to use the cliche, he's the kind of father of protein kinases. And then recent years, he also moved to discover the phosphorylation of other amino acids like histidine. Tony, of course, extremely famous. He is U.S. Academy member of Science and also the American Philosophical Society, member of the U.S. Arts and Science, and also the U.S. Academy of Medicine as well. He's also the Royal Society Fellow of London. Okay, so today's talk is really interesting because I heard part of it somewhere. He's going to talk about how he discovered phosphorylation on tyrosine, and these early works are very, I find it very, very remarkable that he was able to did all this work and have cell paper in a few months and things like that at that time, and very difficult work. And then moving on to using these knowledge, really setting up the whole field of oncogenes, really people able to understand a lot of oncogenes function and now use it to make drugs against all these tyrosine kinases that are really the cornerstones in most of the modern cancer therapies, targeted therapies. So without much ado, please welcome Tony.
Thanks, Randy. It's a great pleasure to be back here again. I think this is my 4th visit to the university, and I've really enjoyed my week here, despite the events going on in Hong Kong. And you're probably wondering about the title, and I'm going to explain, as Randy said, how we've stumbled across tyrosine phosphorylation and how we've made the journey all the way to phosphorylation of histidine. So I thought I'd talk about the history here because it's the 40th anniversary of the discovery of tyrosine phosphorylation. And it all started with this autoradiogram here. So probably many of you in the audience don't know what an autoradiogram is. It's a technique that's pretty outdated now, in which you take a sheet of X-ray film and you place it against a gel or a plate, chromatography plate, and then leave it overnight or some length of time. And this is a method of detecting radioactivity, particularly P32 radioactivity, which is seen when you develop the X-ray film the next day. And so this film developed on June the 14th, 1979, so just over 40 years ago. Was really the start of this whole odyssey. And you probably can't see anything on this, but with the eye of faith, there is a faint spot right here. And I will tell you, how I discovered what this is and what it's led to. So at the time, in 1979, here I am with a longer, slightly darker beard. I was working on polyomavirus, which is a DNA tumor virus that causes tumors in rodents, mice, hamsters. It's a very small circular DNA genome of around 5 kilobases. It encodes 6 genes, 3 of which are expressed as soon as the virus infects a cell. And these 3 proteins are known as T antigens or tumor antigens. They're made actually through overlapping reading frames and alternative splicing. And the most important one is this so-called middle T antigen, which is capable of transforming fibroblasts into cancer cells, at least in culture. And so we were interested in understanding how this middle T antigen converts a normal cell into a cancer cell. And we'd been characterizing these proteins and how they were made, but we had no clue as to mechanism until Mark Collette and Ray Erickson, who've been working on Rous sarcoma virus. This is an RNA tumor virus that also converts fibroblasts into cancer cells. And they reported that the transforming protein of Rous sarcoma virus, that's called VSarc, has an associated protein kinase activity. Now that was exciting because we knew that protein kinases could alter the function of proteins reversibly by phosphorylation. And so we thought maybe this is a universal mechanism of viral transformation. And so we tested whether the polyomavirus middle T antigen has a kinase activity. And we did this by isolating the T antigens using antibodies made against tumor cells and taking the immunoprecipitate and washing it and then incubating it in a kinase buffer with gamma P32 labeled ATP to see if something got phosphorylated. And so on the left here, you can see a gel we ran out of proteins from polyomavirus infected cells labeled with S35 methionine. So that labels the proteins, or with P32. And then in lane C here is the in vitro kinase reaction with gamma labeled, P32 labeled ATP. And much to our delight, we saw a strong P32 label band at the same size as the middle T protein here. And we had a good control here was a set of mutant viruses that were unable to transform cells where there was no signal from, no P32 signal in vitro from their middle T antigen proteins. So there was a correlation between the kinase activity and the ability of these viruses to transform. So here's the middle T band. So we weren't the only groups who had thought of doing this experiment. There was a group in London headed by Alan Smith and a group in Harvard led by Tom Benjamin who'd done similar experiments. And in the summer of May of 1979, we all met up at the Cold Spring Harbor Symposium on tumor viruses. It was the big meeting of the year. We presented our data on the kinase activity associated with the middle T. We discussed with one another how to publish these data and agreed to co-submit our papers to Cell when we all got back, and we submitted our paper on June the 11th, 1979. So basically, we're just reporting there was a kinase activity. So here are the reviews we got on our paper. It was just as hard to get papers into Cell then as it is now. Here's reviewer 1. These two findings force one to seriously consider the possibility that this activity is an artifact. So not a great start. Reviewer 2, reviewer 2. The product of this in vitro protein kinase-like reaction wasn't characterized, and that was true. But with a very low activity, it's possible it's due to some contaminant. And reviewer 3 was most damning of all. Unfortunately, very few of these conclusions drawn by the authors are actually clearly substantiated by the data. So you would have thought this would have meant outright rejection, but luckily, at that time, Ben Lewin was the editor of Cell. He was the founding editor of Cell, and he made his own decisions. He didn't let the reviewers make the decisions. And so he sent, I think we all 3 papers got very similar reviews. They were sent back to us with a letter saying, please revise the paper and resubmit. So I had already guessed before we got the, did it say when the reviews came back? Yeah, we got the reviews back on July the 9th. So I had already guessed that probably one of the questions would be, which amino acid is getting phosphorylated? Is it serine or threonine? It had to be one of the two. And so on June the 12th, so the day after we submitted, I set out to do that experiment, which involved labeling middle T in the same kinase reaction with P32, and then cutting out the band from the gel, hydrolyzing it in 6 normal HCl at 110 degrees for an hour to release amino acids, and then resolving that material with hydrolysate on a thin layer plate by electrophoresis at pH 2. And that's what all this page from my notebook shows you exactly the experiment that I did that day. And so you already saw the autoradiogram that I got. And so what I'd done was to take the dried acid hydrolysate, mixed in some unlabeled phosphoserine and phosphothreonine, and separated these samples on a thin layer plate by electrophoresis. And PS here stands for phosphoserine, and PT stands for phosphothreonine. So that's what I expected. One of them will be labeled, or maybe both. But as you've already seen, the faint spot I saw here didn't migrate with either of them. And on a longer exposure, now you can believe me, there really was a spot there. Here it is, it's quite dark. That's 4 days of exposure with a screen. And you can see it's quite clear that whatever it wasn't phosphoserine or phosphothreonine. So first thought was, well, maybe this is an artifact. Maybe I did something wrong. And so I noted in my lab book here that spot does not coincide with phosphoserine or phosphothreonine. So when you find something like this, the next thing to do is to repeat it, right, just to see if it was an artifact. And so I did the same experiment again on June the 24th and got the same results. So it wasn't an artifact, so it was something that needed to be explained. Now, luckily, I was trained as a biochemist, and so I knew there was a third hydroxy amino acid, and whatever this was, it had to be stable to acid hydrolysis, so it was likely to be a phosphate ester. And so I knew that in addition to serine and threonine, there was tyrosine. No one had reported tyrosine phosphorylation, but it was possible. And so what I was proposing then was that in addition to phosphorylation of serine on this hydroxyl group and threonine on this hydroxyl group, that one could phosphorylate the phenolic hydroxyl group of tyrosine. So how was I going to prove that? Well, I needed some phosphotyrosine marker. Now, you couldn't go out to Sigma and buy it, unfortunately. So I had to try and make my own, and I'm not a chemist, but so I rather naively mixed together phosphorus oxychloride and tyrosine in water, and as you might have predicted, you get a black tar. Luckily, however, I could extract a little soluble phosphotyrosine, something I could characterize as phosphotyrosine, and when I ran it out on July the 2nd, on a thin layer electrophoresis, I found a faint ninhydrin staining spot that migrated between phosphoserine and phosphothreonine. So potentially on the verge of something very exciting. A group of us set out to raft a river in Idaho, so we had to drive 1,100 miles to raft this river with a group of friends. The Salmon River, and from there I flew straight from Salmon, Idaho to London to attend the DNA tumor virus meeting being held in Cambridge. And so I didn't get back for a whole month with this potentially important result. And here I am, rafting on the Salmon River. This is me here with my beard and a couple of passengers here. Anyway, when I got back, we were able to devise a two-dimensional separation for phosphotyrosine using this new marker I had made. And it was pretty clear that when you hydrolyze middle T, phosphorylated in vitro, you got phosphotyrosine and not phosphothreonine or phosphoserine. So this was the figure that went into the revised version of the paper that we sent back. And the paper was submitted on September the 25th. It was accepted on September the 27th. Now remember, this wasn't electronically submitted. This was sent by FedEx from La Jolla to Cambridge in Massachusetts. And so it was clearly accepted the moment it arrived by Ben Lewin, who, as I said, made his own decisions without re-review. And it was published in December that year. Here's the paper. The other two papers were published back-to-back with ours, but either they weren't asked to find the phosphoamino acid or they couldn't do it, and so ours was the only paper that said that. Now, Subsequently, we discovered that the reason that I'd been able to find phosphotyrosine is that I was too lazy to make up fresh pH 1.9 electrophoresis buffer. And if you reuse the buffer over and over again, as we would do, turns out the pH drops to 1.7. It was this subtle difference in pH that led to phosphotyrosine separating from phosphothreonine. And in fact, Ray Erickson's group had reported that another kinase was a threonine kinase when it turned out to be a tyrosine kinase. And then subsequently, Sarah Courtney showed that the polyomavirus-associated kinase activity is not an intrinsic kinase activity, but due to association with the cellular homologue of the viral Salk gene. But there was a second stroke of luck here, because I had a postdoc in the lab, Karen Beeman, who was trained to work on Rous sarcoma virus. And so we had the Rous sarcoma virus system in the lab. So as a control for something I was doing with middle T, so this was on the 18th of September 1979, but right before we resubmitted the paper, I ran out a kinase reaction with the viral Salk protein, which phosphorylates actually the heavy chain of the antibody, hydrolyzed it, and ran it out as a phosphothreonine marker, because that's what Erickson had reported. And much to my amazement, this turned out to be phosphotyrosine as well. So here we now had two very different tumor viruses, each utilizing tyrosine phosphorylation. And so we quickly followed up on the work with the Salk kinase. And shown here is a two-dimensional separation showing that the Salk kinase phosphorylates tyrosine. And most importantly in this case, we were able to show that cells, chick cells, transformed by infection with Rous sarcoma virus into cancer cells contained a strongly elevated amount of phosphotyrosine in total cellular protein. So here's a normal cell. This is a different type of two-dimensional separation. And they have a very low level of phosphotyrosine in protein compared to phosphoserine and phosphothreonine. You can see it's about a 10-fold increase. And we knew from using temperature-sensitive Rous sarcoma virus mutants that this was due to Salk kinase activity. So this pretty much established then that Rous sarcoma virus transforms cells by promoting tyrosine phosphorylation. And in fact, the Salk protein itself is phosphorylated on tyrosine, and we discovered one cellular protein in these cells, CDC37, that's also phosphorylated on tyrosine. So this was published in PNAS in March of 1980, communicated by Bob Hollier, an Academy member in the next door lab. And this was communicated to PNAS in December, but it wasn't published until March. Nothing happened online then. So amazingly, all the experiments in the Cell paper and the PNAS paper were done in less than 5 months from start to finish. And all of those in the PNAS paper were done in less than a month. So you could actually still do things pretty fast 40 years ago, even though we don't have all of the modern kits and technology. So these discoveries led to my interest in protein kinases and protein phosphorylation. And the first sequences of protein kinases were being reported at about this time in the early 1980s. And particularly a number of the viral oncogene products were oncogenes were sequenced because the viruses basically had cloned the sequence for you. And so we knew the sequences of several tyrosine kinases. These were all reported subsequent to be tyrosine kinases subsequent to our work. And there were also some sequences of serine threonine kinases, particularly the cyclic AMP-dependent protein kinase, which was deduced the old-fashioned way. In other words, the protein was sequenced and not the cDNA. And I did these alignments by eye. BB here stands for before blast, so there was no computer program to align sequences. You had to do it yourself. And what I noted were that there were a series of conserved motifs, not only among the tyrosine kinases, but also in the serine kinases as well. And it turns out that this motif, HRDLRAAN, distinguishes the tyrosine kinases from the serine threonine kinases, which have KPEN. So there are 12 motifs, and they're separated along the length of the catalytic domain. Now, the first protein kinase alignment that we did was not published in a regular journal, but rather in the Scientific American, in an article I wrote about the proteins of oncogenes. So this is probably not the place you would have looked for a protein kinase alignment. Now, as I said, there are 12 of these conserved motifs in alignments that we did with Steve Hanks. So this was now nearly 85 protein kinase sequences. And as I said, they are ordered along the length of the catalytic domain, which is around 300 residues. But this catalytic domain can be part of much longer proteins, like the EGF receptor. And subsequent work from Susan Taylor, this was 1991, showed that most of these motifs line the catalytic cleft of the protein kinase catalytic domain, which has an N-terminal lobe and a C-terminal lobe. And this molecule of pKa has a bound ATP in it. And you can see this is the gamma phosphate that's going to be transferred onto a substrate. And so here's that RDL motif. A mutation of any one of these conserved motifs blocks kinase activity. So not only were they conserved at the sequence level, but they're functionally very important. So using alignment based on these motifs, it became possible to count the number of different protein kinases that an organism could express. And you can see this timeline here. So the first kinases were activities that were purified and then sequenced. And Salk was discovered in 1979 as a tyrosine kinase. But you can see how rapidly the number of sequences that were predicted to be protein kinases was rising in the early 1980s. All of this due to cDNA cloning, and particularly PCR-based cloning using primers that were based on those conserved motifs. So at the time, I predicted there might be as many as 1,001 protein kinase genes in a mammalian genome. It turns out that was a slight overestimate. Our current number is around 535. But there are organisms that have much larger numbers of protein kinase genes. For instance, the eucalyptus tree has 2,500, and I think the current champion will be rice, which has nearly 4,500 protein kinase genes. Now, in plants, there's been several rounds of whole genome duplication, which is why there are probably so many kinase genes. So protein kinases are a very common gene type, and together the protein kinases and the phosphatases occupy close to 3% of all genes in a typical eukaryote. So when the human genome sequence was reported in 2001, Gerard Manning and colleagues and I were able to enumerate the number of protein kinase genes in the human genome, and we called this collection of kinases the kinome, and we found 518 protein kinases in total. Most of them can be mapped to a single tree using a sequence similarity. And separated into 7 major types: the tyrosine kinase-like, the basophilic kinases like protein kinase A, the calcium-regulated kinases, and then the proline-directed kinases, including the CDCs and MAP kinases. In addition to these protein kinases, there were about 40 atypical protein kinases that are mostly very distantly related in sequence to the main family. But particularly important are these PI kinase-related kinases, ATM, ATR, mTOR, and DNA PK. So there are 518 kinases in our initial estimate. About 10%, interestingly, are missing key catalytic residues and are classified as pseudokinases, which seem to lack catalytic activity for the most part, but are often conserved and probably have important scaffolding functions. If we move ahead now another 16 years to human kinome 2.0, we've risen a little bit to about 535. This is Pryor's estimate here. All of the new kinases, except for one, I think, are atypical or remote kinases. They include enzymes like FAM20C, which is a secreted kinase. That's the real casein kinase. This phosphorylates secreted proteins like casein. And also the NME family of histidine kinases that I'll talk about. About, or more than a third of these protein kinases have been implicated in disease, particularly in cancer here by gain of function or loss of function mutations. And particularly their role in cancer, starting with Rous sarcoma virus, the Salk kinase, led to a lot of interest in developing small molecule inhibitors of activated tyrosine kinases like Salk. And as of last month, out of the 64 kinase inhibitors that have been approved as cancer drugs, 37 are tyrosine kinase inhibitors. And here's a timeline. This began with the approval of Gleevec, imatinib, in 2001 as a treatment for chronic myelogenous leukemia, which is driven by a fusion mutant form of the Abl tyrosine kinase. And this drug is a very effective treatment. If it's used in the early phase of the disease, it can be an almost a cure. And there are people who went on Gleevec in 2001 who are still alive and still taking Gleevec. So that was my little bit of history, which went on a little bit long, but I'll now switch to our own work, 2 projects. The first on histidine phosphorylation and the second a short story on pancreatic cancer. Now, most of you will know about serine, threonine, and tyrosine phosphorylation. It's what I've been talking about. But what you may not realize is that in addition to those 3 hydroxyamino acids, histidine, arginine, and lysine, so that's the basic amino acids, cysteine, and the 2 acidic amino acids, glutamate and aspartate, can all be phosphorylated. And the reason that we know much less about these phosphoamino acids is that in all cases, these are chemically unstable residues, and so they're much more difficult to study. So for over 40 years now, I've been interested in studying tyrosine phosphorylation where the phosphate is linked to the phenolic hydroxyl group, creating a phosphate ester, which is very heat stable. In fact, you can boil an acid, as I showed you, to release it from proteins. But about 25 years ago, I became interested in studying histidine phosphorylation because in some ways it's sort of analogous to tyrosine. It has a ring side chain, in this case an imidazole ring. And we already knew that it could be phosphorylated in enzyme intermediates like phosphoglycerate mutase. And histidine is unique in that it can create 2 different phosphoisoforms. So it can either be phosphorylated at the N1 position or at the N3 position. And in both cases, you're creating a phosphorus-nitrogen bond, which is a phosphoramidate linkage. And these such bonds are heat and acid labile, so you cannot hydrolyze the protein in acid and release phosphohistidine, for instance. So what do we know about histidine phosphorylation? It was first discovered in a mitochondrial enzyme by Paul Boyer in 1962, so it has a longer history than phosphotyrosine. And if you know anything, it's probably its role in bacterial signaling through the two-component systems, where a receptor-like molecule that has an extracellular domain is activated in response to a signal, often a small molecule, that results in autophosphorylation on a histidine in the receptor, which is then transferred onto an aspartate residue in a response regulator, commonly a transcription factor. So here you have two non-canonical phosphorylation events in series here. And it's this phosphorylated protein that delivers the biological output. Now, this type of system is not found in metazoans, in multicellular organisms. Instead, we have sort of conventional protein kinases, but there are also enzymes reported to be histidine kinases, particularly this NME1 and NME2, which autophosphorylate on a histidine, and that phosphate can be transferred onto a protein. And there are several enzymes that have phosphohistidine enzyme intermediates, including NMEs, phosphoglycerate mutase, ATP citrate lyase, phospholipase D are examples. And the reaction that NME normally carries out is to act as a nucleoside diphosphate kinase, where the phosphorylated form of NME, the intermediate, phosphorylates, it's phosphorylated via using ATP, but the phospho form then donates its phosphate to a nucleoside diphosphate, let's say GDP recreating GTP. But there's evidence that that phosphate can also be transferred onto a protein at a histidine residue to form a phosphohistidine-containing protein. And so we know of a number of proteins that are phosphorylated on histidine. This potassium channel here is opened by phosphorylation of histidine 358 in the C-terminal tail. And it's been known for many years that histone H4 is phosphorylated on a histidine in its N-terminal tail, although its function is still unclear. So histidine phosphorylation dependent processes are known in many parts of the cell. We have these ion channels, we have G proteins that are histidine phosphorylated. We know there's histidine phosphorylation events in the mitochondrion. And in fact, there's a specialized form of NME, NME4, that is found in the mitochondrion. There's also histidine phosphorylation at the centrosome, a specialized form, NME7, that Bob Chi has worked on here. And then there's histidine phosphorylation in the nucleus. So about 25 years ago, we thought it would be interesting to try and develop antibodies to phosphohistidine by analogy with the antibodies that have been so useful in studying tyrosine phosphorylation. And we tried to raise antibodies to phosphohistidine, but failed, probably because it was too unstable in the animal. And so we had to wait until someone devised non-cleavable stable analogs of phosphohistidine, and these phosphoryl triazolyl alanines were developed by Tom Muir at Princeton and Mike Webb at Leeds. And these are phosphorus carbon bonds, so they're phosphonates, totally stable to hydrolysis. And they have 5-membered rings that look a little like a histidine, an imidazole ring, but really not a great analog. And so what we set out to do was to try and develop sequence-independent antiphosphohistidine antibodies by incorporating these analogs into a degenerate peptide backbone containing random alanine and glycine residues. And these peptides were coupled to KLH and then used to immunize rabbits. And we got out antibodies with the expected properties. So for instance, the antibodies raised against 1-PTZA recognize NME1. This is the active phosphoenzyme form here, and not phosphoglycerate mutase, which is recognized instead by the 3-phosphohistidine antibodies. Now, I told you couldn't heat phosphohistidine because you lost it. Well, these gels are actually run without heating the sample in sample buffer, and the gels are actually run in the cold room to preserve the phosphohistidine. But if you do boil the sample here, this sample here was boiled, you can see most of the phosphoglycerate mutase signal goes away here. So we went ahead and used the spleens of these rabbits to generate monoclonal antibodies against 1-phosphohistidine and 3-phosphohistidine. And you can see here's one of the 1-p-His antibodies used to blot a series of cancer cell lines. In every case, there is a heated control lane, which is boiled in sample buffer, and an unheated lane. And you can see the major bands actually NME1 and NME2 themselves. There are some other bands that go away with heating. If we use 3-phosphohistidine, two of the different monoclonals here, on let's say 293 cells, you can see a lot of bands that go away with heating. So we think it's likely that 3-p-His is more prevalent than 1-p-His in cells. And you can see a lot of bands in E. coli, 3-p-His bands in E. coli that go away. So altogether, we have 3 different 1-p-His antibodies and 4 3-p-His antibodies. They're relatively, but not totally sequence independent, I would say. And we can use these for immunoblotting, which I've already shown you, for IF staining, for immunohistochemistry, and as I will show you also, affinity enrichment of p-His proteins or peptides for identification. We've also got structures of most of these monoclonal antibodies bound to phosphatase containing peptides to define how they recognize 1 and 3-p-His selectively. So here's one of the 1-p-His monoclonals binding 1-p-His. You see the 1 position here. Contacts actually both with the heavy chain CDRs and the light chain CDRs. Very little contact with the backbone, so that explains why they're sequence independent. Here's one of the 3-p-His antibodies contacting, a lot of contacts with the phosphate at the 3 position, very few with the peptide backbone. And if you compare, just look on the top right here, you compare the orientation of these peptides when they're bound to their antibodies, they're quite different, and there would be steric clashes. So a 1-p-His antibody can't bind 3-p-His and vice versa. So when we use these antibodies to stain cells, in this case proliferating HeLa cells, and this is a 3-p-His antibody, you see some nuclear stain, you see some green cytoplasmic stain, and you also see this dot in the cytoplasm. In early prophase, this dot seems to duplicate and it becomes the spindle pole. So what we're seeing here is 3-p-His staining of something in the centrosomes and in the spindle poles. So that suggests there may be a function for histidine phosphorylation in the cell cycle. We can also use these antibodies to define whether a protein is phosphorylated on the 1 or 3 position. So here's an example in work we did together with Ed Skolnick's group of this potassium channel, the calcium-activated potassium channel, that in this case is expressed in Jurkat T cells stably with a GFP tag. So immunoprecipitating this with anti-GFP antibodies and then blotted for 3-p-His or 1-p-His. You can see that the protein contains 3-p-His but not 1-p-His. And then conversely, NME2, one of the histidine kinases, contains 1 p-his and not 3 p-his. So we actually know a little more about what the phosphorylation of this channel might do. So this is histidine 358 in the tail, and work from Ed Skolnick and Steve Hubbard suggests that the phosphate on the N3 position of this histidine is blocking the binding of an inhibitory copper ion, which holds the 4 tails. So this is a tetrameric channel, like most potassium channels. It holds the 4 cytoplasmic tails of this channel in an inactive configuration. The channel is activated by calcium. This binds to the calmodulin that's already resident on the channel. And this then allows that histidine to be phosphorylated by NME2, and the phosphate on the histidine now kicks off the copper or prevents copper binding. And so potentially then this is one of the functions of histidine phosphorylation, is to regulate metal ion binding. We also used immobilized monoclonal antibodies to pull out proteins from, in this case, 293 T cells, lysed at pH 10 to stabilize the phosphohistidine and 6 molar urea, which was to dissociate any protein complexes. And then we compared the proteins that bound to these immobilized antibodies from the lysates, either with or without treatment at pH 6 and boiling. And the proteins that specifically eluted with a 2-fold enrichment are listed here, or the GO terms are listed here. We found around 780 proteins. Many of them seem to be involved in processes that regulate RNA biology. A significant number were reported to be cell cycle regulated. And we found phosphohistidine containing proteins that we expected, histone H4, NME, PGAM, and ACLY. Now, this doesn't directly show these proteins are histidine phosphorylated, and for that we need to map the sites of histidine phosphorylation. And this is challenging because, as I already told you, phosphohistidine is labile in acid, and most methods for doing phosphoproteomic analysis use strong acid to enrich and to separate the peptides for mass spec. So we tried to develop methods that were gentler, particularly enriching the phosphopeptides at pH 8 using, in this case, immobilized antibodies or hydroxyapatite chromatography. And then we eluted at pH 11. Most antibodies denature at pH 11. But phosphohistidine is stable, and then use a slightly modified buffer for the HPLC step. So this is the work of Kevin Adam. And using just, in this case, immunoaffinity enrichment. And then searching the spectra of the peptides that were bound and released for phosphoserine, threonine, tyrosine, histidine, lysine, or arginine. We found a number of phosphohistidine sites. Interestingly, we also found some phospholysine and phosphoarginine sites that we didn't expect. For some reason that we don't really understand. Our antibodies also select these peptides. They have phosphoramidate bonds, so that may be the reason. So we have tentatively identified a number of new phosphohistidine sites. Here's one phosphoglycerate mutase that we already knew, but it's a nice control. And here are just a few of the potentially interesting sites that we found. For instance, LCK, the Salk family tyrosine kinase. It would be this histidine that's phosphorylated. Here are some phosphoarginine sites, another one in a kinase, ATM here. Here are some phospholysine sites. When an arginine or a lysine is phosphorylated, trypsin can no longer cleave it. So that's a criterion for phosphoarginine and phospholysine sites. There's no obvious motif for the phosphohistidine or at the other sites. But if you do one of these logos, you can see we find a lot of leucines around the histidine on average, and this is a compilation, right? And we wonder whether the leucine environment actually is protecting the histidine from hydrolysis because it's very hydrophobic. So that could be we're enriching a subset of the phosphohistidine peptides. Here's another set with lower probability, largely because the peptide has histidine, lysine, and arginine residues, and we can't really tell which is being phosphorylated. So other people are trying to do the same thing, particularly Claire Ayer's group at the University of Liverpool has developed a neutral method for enriching non-canonical phosphopeptides using strong anion exchange. It's called UPAX. It was just published in Embo Journal in August. And she's identified a lot of phosphohistidine, arginine, lysine, aspartate, and glutamate sites in HeLa cells. And there are other people who are developing methods for phosphohistidine peptide enrichment. So all of this leads us to suspect that the so-called hidden phosphoproteome, as we call phosphorylation of these difficult to study phosphosites, could be quite large, maybe as much as 20% of the phosphoproteome. So there could be a lot of biology hidden here that we haven't really suspected because we can't really analyze these sites. So clearly what we need to do is to take some of these sites, validate them by direct mutagenesis to show the signal goes away. We can then make phosphose-specific antibodies against that site. We've done that for NME1 and NME2, so we know we can do it to really study when and where and how they're phosphorylated. So there are a lot of open questions. How big is the phosphohistidine proteome? Phosphoarginine, phospholysine? Are these proteins phosphorylated at the 1 or 3 position? Mass spec can't tell you which it is. Under what conditions is phosphorylation of these sites regulated? How important are the NMEs as the kinases? Are there other kinases? Are there conventional kinases that might be able to do this? And what are the phosphatases? I'll talk about phosphatases in just a second. How can one study this genetically? One can mutate the histidine, and generally it's replaced with asparagine because it's isosteric, but asparagine isn't charged. So it's not a great non-phosphorylatable substitution. Perhaps more importantly, how can we make a phosphomimetic mutation? And we really can't. So glutamate and aspartate won't work here. So we're trying to use our natural amino acids, using the expanded genetic code to install a non-cleavable 1 or 3p his analog in place of the histidine. And we're doing this together with Lei Wang at UCSF. So what could histidine phosphorylation be used for? Is it simply short-term responses because it's unstable chemically even in the cell? Are there phosphohistidine binding proteins? Could this work through local charge changes? You go from +1 to -2, that's a big change. And does it work in other cases by regulating divalent metal ion binding? So there's plenty to do here. We've got a lot of, we've made some new reagents and there are new technologies. So I think this area is ripe for further investigation. Now, before I switch to pancreatic cancer, I'll just tell you one very quick story of how we've used these antibodies to study a biological process in collaboration with Mike Hall's group in Basel, who had generated a mutant mouse where they could conditionally knock out P10 and TSC1 in the liver. And these combined mutations lead to an increase in mTOR pathway signaling. Knocking out TSC1 increases mTORC1. Knocking out P10 increases mTORC2 and mTORC1. And these mice develop a very rapid and fulminant hepatocellular carcinoma within about four months here. You can see it's multifocal, which is interesting. And they set out actually to study these tumors not using transcription analysis, but rather proteome analysis, looking particularly for protein kinases and phosphatases whose proteins were up or down regulated. And what they noted was that the highest upregulated protein kinases were NME1 and NME2, and the phosphatase that was most strongly downregulated was LHPP. And what I forgot to point out in an earlier slide is that there are 3 phosphohistidine phosphatases that have been reported: LHPP, PHPT1, and PGAM5, a distant relative of the glycolytic enzyme. And so we got together with Mike to look to see whether the increase in the histidine kinases and the decrease in the histidine phosphatase might affect histidine phosphorylation in these tumors. We would have predicted an increase, and that's what we found, particularly the work of Sravant Hindupur, that there's an increase in the level of 3 and 1 p-his in the tumor samples compared to normal liver. So each case here, there is the unboiled and a boiled sample. And there's about a 2 to 3-fold increase in both 1 and 3 p-his proteins. And they were able to show by expressing LHPP using adenovirus in a cell line derived from one of these tumors that high levels of the phosphatase blocked the ability of these cells to form hepatospheres in culture. And particularly important, re-expression using AAV injection into the tail vein caused a great decrease in the number and size of tumors that were formed in these mice. You can see a dramatic decrease here. So this suggests then that LHPP, by inhibiting histidine phosphorylation, is acting as a tumor suppressor. Now that's in mice. What about in humans? And they collaborated with a group of oncologists in Basel to obtain patient hepatocellular carcinoma tumor tissue. And here are 2 patients showing an increase in the level of 3 phosphohistidine proteins compared to the normal adjacent liver. Again, it's about a 2 to 3-fold increase. So this suggests then that LHPP is a phosphohistidine phosphatase, acts as a tumor suppressor. Patients, in fact, with low LHPP RNA levels have a worse prognosis. And what we need to do now is to characterize which histidine phosphorylated proteins are elevated. And in the paper, there were a few studies of that sort to see if we can really define the mechanism through which histidine phosphorylation might drive liver cancer. And the long-term hope is that one might be able to target either the histidine kinases themselves or some process downstream of histidine phosphorylation as a new type of therapy. Okay, I'm going to take 10 more minutes to tell you the second story. That's about right, yeah. Which is a total change in direction and involves pancreatic adenocarcinoma, which is one of the most lethal cancers. It's the 3rd leading cause of death right now with the highest mortality rate. It's a silent tumor. It develops almost without any symptoms. And it's usually ultimately diagnosed too late when the tumor has already metastasized. So we know quite a lot about the progression of this disease. It has a characteristic series of genetic changes that drive different morphological or pathological changes. So often a KRAS activating mutation is early. Loss of P16F is subsequent, and then finally loss of P53 and SMAD4. And these correlate with the formation of 3 different morphologies called PANINs, pancreatic cancer in situ, as it were. And PANIN3 then rapidly progresses into a more aggressive and invasive tumor, which can lead to extravasation into the, intravasation into the bloodstream, extravasation, and the formation of a distant metastasis. Now, like all solid tumors, pancreatic cancer isn't just tumor cells. It contains many other normal cell types. So here we have the tumor cells in gray here, but there are blood vessels that are important for delivering oxygen and nutrients to the tumor. There are many types of infiltrating immune cells, B and T cells and macrophages particularly. And there are also cancer-associated fibroblasts or mesenchymal cells of various sorts. And I'm going to focus on a particular subset of these called stellate cells. So in fact, the tumor is really a tissue. It's a community of cells that are interacting with one another. And so when one thinks about tumorigenesis and therapy, one needs to consider what these normal cells are doing in the tumor. Now, stellate cells are a particular type of mesenchymal cell found in the pancreas, in the liver, and in the kidney, and they are characteristically star-shaped, hence the name stellate cell. It's been estimated that about 4% of all cells in a normal pancreas. They're quiescent, they sit there, and their main role is to surveil the tissue for damage. And in response to a tissue injury or inflammation, they transform from this quiescent state into a myofibroblast-like state. So they change shape, they start to proliferate, and they begin to secrete a lot of things, including extracellular matrix and cytokines. And these cells really sort of wall off an incipient tumor. So they form this massive sort of shield around the tumor cells. It's really an injury response. That's what would happen in an injury. And so actually this so-called reaction really sort of makes the tumor cells inaccessible to blood vessels and actually to drugs. And so that's one of the challenges in treating pancreatic cancer is to get treatments to the tumor cells themselves. So about 8 years ago now, I became part of a Stand Up to Cancer dream team focused on pancreatic cancer, and our goal as a team at the Salk Institute was to study the role of the stellate cell in pancreatic cancer, and particularly ours was to look to see what factors do these stellate cells make that might act on the tumor cells and vice versa. So what we were proposing then was that the tumor cells themselves make soluble factors like TGF-beta and PDGF that activate the quiescent stellate cells. They are converted to myofibroblasts, which then secrete a lot of factors, soluble factors, paracrine factors, that we would argue then would support tumor progression. And what I'm going to tell you is that we've identified LIF, or leukemia inhibitory factor, as a cytokine secreted by activated stellate cells that supports tumor cell progression. So we started, and this was the work of Yu Shi, a postdoc in my lab, and his friend and collaborator, Rous Tian, who is now at SUSTech in Shenzhen, but at the time was a postdoc in Tony Paulson's lab. And they decided to look at the global spectrum of proteins secreted by the tumor cells. So this is a human pancreatic tumor cell line, MIA-PACA2, and cultured stellate cells. And then survey this list to see if one could find something relevant. Now, I would agree that these cells were grown on plastic, two-dimensional, regular medium. This is not really reflective of what happens in the tumor, but it was the place we started. And these secretomes are very complicated based on mass spec analysis, 2,400 proteins in the MIA-PACA2 secretome and 1,400 in the stellate cell secretome. And we found about 400 proteins uniquely secreted by these tumor cells and 170 by the stellate cells. So what do we find? So we were particularly interested in growth factors and cytokines, which could act in a paracrine fashion. But I should point out that the stellate cells do make massive numbers and amounts of extracellular matrix proteins and extracellular matrix remodelers that are not made by the tumor cells. So this is the desmoplastic reaction. Both cell types make a lot of growth factors and cytokines. And for instance, the tumor cells make PDGF, which activates the stellate cells. They also make TGF-beta. But we sort of focused on the IL-6 family of cytokines, and particularly LIF, for reasons I'm going to tell you, LIF, leukemia inhibitory factor. So why do we focus on LIF? Well, we noted that the conditioned medium from the stellate cells stimulated phosphorylation of STAT3 in the tumor cells, and STAT3 is downstream of most cytokines. We found that the conditioned medium elicited binding of STAT3 to the LIF receptor and not the IL-6 receptor, for instance. And we showed that a neutralizing anti-LIF antibody abolished most of the ability of the conditioned medium to stimulate STAT3 phosphorylation. LIF is a stem cell factor. People use it to maintain ES cells in their pluripotent state. And it can also elicit an invasive phenotype. It's used by the blastocyst to embed in the placental wall. So it creates an invasive state. So that was interesting from a perspective of tumor cell invasion. We noted that LIF RNA levels are highest in pancreatic cancer among 20 human tumor cell types that were studied by the TCGA. You can see there's very low levels in the normal pancreas and very high levels in tumor cells. So that also piqued our interest. And high LIF RNA levels correlate with poor tumor prognosis. So LIF is a member of this three-member family, IL-6, IL-11, and LIF. They all use a common signaling subunit, GP130. They each have a unique receptor subunit, so an IL-6 receptor, an IL-11 receptor, and a LIF receptor. And these cytokines signal through JAK-STAT signaling primarily, and JAK-STAT leads to phosphorylation of STAT3 at tyrosine 705. Now, we noted that IL-6, IL-11, and LIF are all secreted by human stellate cells in culture, but we focused on LIF because we could show that knockout of the LIF receptor in this KP4 human pancreatic cancer cell line reduced STAT3 phosphorylation by conditioned medium pretty strongly, and the use of a neutralizing antibody also almost completely abolished the conditioned medium's ability to stimulate STAT3. So we've sort of not looked at IL-6 and IL-11, and there are other reasons why we didn't follow them up, but LIF is something that then we wanted to follow. And we needed now to begin to test whether LIF could serve as a driver in pancreatic cancer. We needed a preclinical mouse model, and we've used the so-called KPC mouse developed by Dave Tuvison. And this mouse has a knock-in mutation into one of the KRAS alleles, which has a stop LOX element and a G12D activating mutation. And these mice are also the ones we used, have both alleles of p53 floxed. And the driver CRE to activate KRAS mutant expression and to knock out p53 is PDX1 CRE, which is expressed early on in the development of the pancreas. And these mice develop pancreatic cancer early, and they have a very rapid disease onset, and they die on average around 50 days after birth. And we could use a conditional allele of the LIF receptor crossed into the KPC mice and show that loss of both alleles of LIF receptor, both floxed alleles, increased survival and increased sensitivity to gemcitabine, a nucleoside analog that's used in treating people with pancreatic cancer. So the LIF receptor in this case is being knocked out in the pancreatic ductal cells, which are the cells that go on to form the tumor, driven by activated RAS and loss of p53. So this was consistent then with LIF being important, driving these tumors. And we could use in situ hybridization using RNA-scope 2 color system to show that the red LIF RNA was mostly expressed in tumors in the stromal cells surrounding the blue tumor cells marked here by keratin 19, an epithelial cell marker, and that the LIF receptor was mostly expressed in the tumor cells themselves, the red LIF receptor, as opposed to in the surrounding blue periostin positive stromal cells. So this suggested then that it was the cellate cells in the tumor that were making LIF. They were acting on the tumor cells, which are LIF receptor positive. We also had some evidence that the tumor cells might also make some LIF. Now that's in mouse, and we see the same thing in the human pancreatic cancer samples. So again, here LIF RNA is red, and you can see it's mostly in the cells surrounding the blue keratin 19 positive tumor cells. So then we had to establish a preclinical treatment model, and we decided to use this neutralizing LIF monoclonal antibody. And we established a dose of 25 mg per kg 3 times a week. And if we treat a mouse, a KPC mouse, and then stain for phosphostat 3, you can see this is the control, IgG. All these nuclei, tumor cell nuclei, are brown for phosphostat 3. And if we neutralize with this antibody, you can see most of them go blue. So this was the protocol we established. We used the LIF monoclonal antibody at 25 mg per kg 3 times a week, and then we used gemcitabine here on this schedule at 50 mg per kg. So I should say we started at 32 days of age after the mice were weaned, but at a time when tumors were already detectable. So this is a treatment protocol rather than a prevention protocol. And so this is the control. The mice die around 50 days on average. If we simply use the anti-LIF antibody, there is an increase, a statistically significant increase in survival, but a particularly strong survival if we combine gemcitabine and the anti-LIF antibody. So that's encouraging. And if we use a slightly different model where we've pre-treated the mice with a chemotherapy regimen that's used in people, paclitaxel, gemcitabine and cisplatin, and then for a week, and then treat with gemcitabine and antibody or IgG, we can see a very significant increase in survival. And so what we conclude is then that LIF blockade with a neutralizing antibody slows tumor progression and increases the response to chemotherapy in this model of pancreatic cancer. So what is LIF neutralization, LIF blockade doing? Well, it's increasing the percentage of the tumor tissue that is moderately or well differentiated, decreasing the area that is poorly differentiated. It's decreasing the number of tumor cells. These are EpCAM positive tumor cells isolated from the tumor that are stem cell marker positive, and it greatly decreases the ability of tumor cells to form tumors in mice, done in collaboration with Nikki Lytle and Tanish Serea. So LIF blockade is reducing the relative abundance of tumor cells with stem cell-like properties, therefore increasing tumor cell differentiation, and that is sort of consistent with what we know about LIF, is it's a stem cell factor. The levels of LIF increase steadily during tumor progression in the mouse, and actually they're also increased in mice with chronic pancreatitis, which activates stellate cells, but not as highly as in the tumors. You can see this is a log scale for LIF here by ELISA. And we can also begin to detect LIF in the serum of these mice at 5 weeks. So that's mice. We have positive results suggesting LIF is serving a positive role in tumor progression. What about in people? So by ELISA assay on human tumor samples, pancreatic tumor samples, we can see many tumors have high levels of LIF compared to the normal pancreas or chronic pancreatitis. So it's quite a wide range, I agree. And we've collaborated with Rijan Tian, again at SUSTEC now, who's analyzed by PRM mass spec LIF levels in a totally different set of tumor samples. And again, there are tumors with very high levels of LIF. On average, there's a significant increase. In addition, we can also detect LIF in the serum or plasma of human pancreatic cancer patients, now using a slightly different ELISA assay. This is the single molecular ELISA assay, the Quanterix instrument, which is 10 to 100 times more sensitive than a regular ELISA assay. And you can see again, many patients have very high levels of LIF in their serum. And if we look at the levels of LIF this is tumor tissue again now, correlating it with disease status as determined by pathologists, we can see that the levels of LIF in the tumor increase steadily as the differentiation status decreases. And there is a trend towards a correlation between LIF levels and survival. Now, the current biomarker for pancreatic cancer is CA19-9. It's a sugar molecule, and it's used to diagnose and to follow treatment response. And so here we've analyzed a set of tumors. This is actually serum level now for LIF and CA19-9, and you can see that there are high levels of LIF and high levels of CA19-9. But some tumor samples that have high LIF have low CA19-9 and vice versa. So we think maybe a combination of LIF and CA19-9 might be used for biomarker analysis. And then one final piece of data. We've collaborated with the group at Honor Health, Erkut Borazanki and Dan Van Hoff, to study a series of 14 patients that were treated with Abraxane, that's nab-paclitaxel, gemcitabine, cisplatin, paracalcitol, and the checkpoint antibody nivolumab over 14 days, and checked LIF serum levels, baseline and post 14 days treatment. And the patients who showed an objective response are shown in green, and most of them showed a decline in the level of LIF. So this is just a first experiment, but it's quite encouraging. It suggests that LIF could be used as a biomarker. So to summarize then, we have evidence that LIF plays a driver role in mouse model of pancreatic cancer, that LIF acts on the tumor cells, but it could also act on other CAFs, cancer-associated fibroblasts, myeloid suppressor cells, tumor-associated macrophages, and even neurons. In the human situation, LIF levels are positively correlated with progression in human pancreatic cancer, and we think it could serve as a biomarker. And the obvious question is now, can it be targeted therapeutically in humans? And Northern Biologics, a company I have no interest in, no financial interest in, has initiated a phase 1 trial with a humanized anti-LIF antibody last year in highly refractory cancer patients, including 9 pancreatic cancer patients. And the results of this trial, the dose escalation trial and safety trial, is currently being analyzed. So let me finish then by thanking the people who did all the work. Steve Foos spearheaded the antibody work against phosphohistidine with help from Jill, Aaron, and Kevin, who did mass spec, Rajasri, who did structure work, and Natalie, whose work I didn't discuss. And the peptides, the PTZA peptides, were made by Jacques Marget's group. And the work on liver cancer was done with Mike Hall, with the potassium ion channel work was done with Ed Skolnick, and mass spec by John Yates's group, Jim Marisco, and Jolene. So we're not giving up on other phosphoamino acids, and we're currently trying to make antibodies to phosphorylation, so stay tuned for that. And then the work on LIF and pancreatic cancer was done largely by Yuxi in my lab and Ruijun Tian at SUSTEC, and an army of collaborators, and it was funded by Stand Up to Cancer, the Lustgarten Foundation, and the National Cancer Institute. Thank you very much.
A couple of questions.
Yeah.
For the pancreatic cells to stimulate surrounding normal cells to do something to feed the cancer cells, is there any evidence of proliferation of the normal cells, or just activation, differentiation, or something?
So the normal stromal cells are mitotic. So the stellate cells are definitely mitotic in this situation. So the tumor cells...
Any enhancement of proliferation then?
Oh, yeah. So there are very few mitotic cells in a normal pancreas, and the stellate cells are quiescent. You see a few, non-tissue cells like macrophages and well, not macrophages, but other cells that are proliferating within the pancreas, but it's really very quiescent, a pancreas. And so the tumor environment is very pro-mitotic, not only for the tumor cells, but also for the surrounding stromal cells, yes.
Okay. So the last question is about the LIF. In addition to monoclonal antibody, do you know of any efforts to develop small molecule inhibitors?
Yes, there was a paper in Molecular Cancer Therapeutics in July describing a small molecule inhibitor of LIF receptor interaction called EC359, and we wrote a small sort of mini commentary on that paper in the same issue. And excitingly, it was co-authored by our oldest son, who is a graduate student at Stanford, who independently decided to work on targeting LIF in pancreatic cancer. So he started that project before he knew that we were working on it.
Oh, so then was it a disruptor of interactions by screening was done by disruption of the interactions, or looking for binders of...
The group who did it, which wasn't our son, he's using a different strategy to generate a LIF antagonist using a fragment of the LIF receptor as an antagonist. The group who did it actually modeled that there's a structure of the LIF receptor complex from Chris Garcia's group, and they modeled the binding site of LIF on the LIF receptor for an interacting molecule. And they found something which they then evolved. And so it was sort of done computationally rather than by screening large numbers of molecules.
Oh, on the last one, was any potential or actual toxicities if you inhibit LIF in the animal models?
We didn't see, so we only treated, remember, for about 30 or 40 days with the anti-LIF antibody. There's no obvious toxicity, but of course we didn't really do a very thorough survey. But in people it seems to be very well tolerated. The LIF knockout mouse is viable. It's sterile but viable, so you probably wouldn't expect very strong toxicities, but of course you can't really predict. In contrast, the LIF receptor knockout is lethal, and we believe that's because the LIF receptor actually participates as part of a receptor for 5 other cytokines. It was on my slide, but I didn't talk about it. So, for instance, oncostatin M uses the LIF receptor as part of one of its 2 receptors. And so if you knock out the LIF receptor, you're knocking out many things. But if you knock out LIF, you're only apparently affecting just this one signaling system. So I think it could be a fairly safe drug, but we'll see.
I think, is there any animal-based evidence showing the requirement of stellate cells for the development of pancreatic cancer?
Right, so you heard the question. It's a bit complicated. If you knock out, so there are 2 groups who've knocked out CAFs in the KPC or equivalent model of pancreatic cancer, and the cancer gets more aggressive rather than less aggressive. So that would argue that they're not required. Apparently, though, that is probably because this desmoplastic response is preventing the cells from escaping, and so if you remove those cells, they can now metastasize, and so the mice die of a different sort of disease. Rod Evans's group has been working on the role of the vitamin D receptor in the stellate cells. And what he has shown, what he published in several papers now, is that if you activate the vitamin D receptor, the stellate cells convert from an activated state into a more quiescent state. And if you treat mice and now people with a vitamin D analog, then you do get some partial inhibition of tumor progression. So that would support the idea that they are needed. So I'm sorry, it's not a simple answer, but that's what it is, yes.
Okay, another question is, are the kinases responsible for arginine phosphorylation, lysine phosphorylation positively identified?
There's almost nothing known about lysine phosphorylation. There was a report about 50 years ago of a lysine kinase activity in a liver extract. But apart from that, nothing has been done as far as I'm aware. For arginine kinases, there's been a fair amount in bacteria. There are arginine kinases and phosphatases in some bacterial species. People have made phosphoarginine antibodies, which are available. So, and there definitely are. We found phosphoarginine sites, so did Claire Ayers, and so did another group. So I think there's going to be a significant amount of arginine phosphorylation in mammalian cells. What the kinases are, we need to figure that out. But now you've got with a phosphoarginine antibody, you can assay for an activity, I guess. And it's even formally possible that some of these phosphorylations are not enzymatic. So there are activated creatine phosphate, for instance, could probably phosphorylate arginine. IP7, which has a pyrophosphate. So we're not totally convinced it'll have to be an enzymatic reaction, but it probably is because there are phosphatases. So.
Since the phosphohistidine is chemically more labile than the phosphotyrosine, so nature may prefer to use the phosphorylation of histidine for some short-term biological process, such as maybe during the cell division or for the neuron, during memory. And do you know anyone testing those possibilities?
Well, it's certainly something we thought about. In fact, it was one of the possible functions that I noted on my slide. It's used that histidine phosphorylation will be used for a short-term response. And suddenly, in the nervous system will be very interesting. Several ion channels do seem to be regulated by histidine phosphorylation, which will be consistent with that idea, although most of that work was done in T cells, but could certainly happen in a neuron. So I think it's an interesting idea, yes. We're not currently working on it, but I think, again, with the reagents that we and others are developing, it should be possible to begin to look at that and identify. So you'd have to map sites and see if there are interesting, parts of, say, neurotransmitter receptors, and then take it from there. But it's an interesting idea, I agree.
Well, in that case, we look forward to Hunter and Hunter paper on and let's become competitor with you. So let's thank Tony again for in front of the talk.